Right, Ji. Today we will be talking about mutations. First of all, we have to define what is mutations, right? What is a mutation? Mutation is basically a permanent change in nucleotide sequence of a DNA. Permanent change, the features of mutations are: number one, it should be permanent. Change in nucleotide sequence. Of DNA. Permanent mean that which is not repaired. Permanent mean which is not repaired. This concept is very important because actually there daily in our every cell lot of damages occur to the DNA, but they are repaired. And if some damage occur to the DNA and that damage has been repaired, then such damages or such alterations which are repaired and or corrected, we cannot call them mutation. Mutation is that there is some change, injury or damage to a part of a DNA which results in the change in the nucleotide sequence of the DNA, and it is not repaired. And very important point, that when cell will multiply or proliferate, or when DNA will replicate, this change will pass to the progeny cells, next generation of cells. This is very important point related with mutation. Just damage to DNA is not mutation, because many times damage occur to DNA, but this damage is repaired. Is that right? So we were talking about what are mutations. Mutations are permanent changes in the nucleotide sequence of the DNA which are not repaired, and without repair these changes are passed to the next generation of cells, right? You can say which are changes in the DNA which are heritable. Heritable mean what? This word is again very important. Heritable mean when cell will replicate its DNA, right, then this altered nucleotide sequence must be passing to the next generation of the cells, right? Now, mutations can occur in two types of cells. Mutations can occur in somatic cells or mutations can occur in germ cells. Germ cells. Now, let us suppose if mutation occur in one of my somatic cell, what will happen? When nucleotide sequence in my somatic cell is altered, right, we will talk later on. If it is a silent mutation, there will be no change. Even though DNA has been altered, the structure or function of the cell will not be altered, or properties of cell will not be altered, right? But if mutation is of so significance that it brings significant changes in the proteins which are produced by the genes, then there will be alteration in the properties of the cells. Now, if there is significant mutation and that mutation results in alteration in proteins of the cell in such a way, right, that cell proteins are significantly altered, what may happen to the cell? Cell will be having abnormal or altered structural or functional properties. Structural or functional properties. The classical example is a normal cell may convert into malignant cell. Normal cell may convert into malignant cell, right? Because normal cell has a normal genes which control the growth of the cell. But if cell growth regulating genes are significantly disturbed, right, then it is quite possible that cells start over proliferating and it start over living, and that may end up into malignancy. Number one. Number two, sometimes it happens that there is a change in somatic cell of a fetus or embryo. For example, there is a lady who is carrying a product of conception in her uterus, and let's suppose in the ectodermal cell or mesodermal cell or endodermal cell, or even at some advanced stage of the development of the product of conception, if there is some mutation, it's quite possible that may end up into congenital defect, right? That may end up into congenital defect. For example, let's suppose when in the fetus fingers are developing, if some cells related with this finger, they undergo mutation and then they don't grow properly, then this finger part will be abnormal, right? So we'll say this was a somatic cell, but it produced anomaly before the birth, right? And this should be considered one of the congenital defects. Is that clear? Again, I will repeat it. What really happens? What are mutations? Mutations are permanent alteration in the nucleotide sequence of DNA, which can be passed to the next generation of cell, right? If it is somatic cell, the next generation of the proliferating cells, and if it is a germ cell, then this change may be transferred to the next generation of that family, right? Now, as I was telling, that mutations may occur in somatic cells or may occur in the germ cell, that is in sperm or ova. If mutations occur in somatic cell, they may occur before birth or after birth. If mutations occur before birth, that may end up into congenital defect, and if they occur after the birth, they may produce cells or mass of cells with abnormal structural and functional properties. For example, that mass may end up into neoplastic change and carcinoma, right? Then we can talk about mutations in germ cells. For example, such type of problem may occur into ovum, or it may occur to the sperm, genetic material of sperm. Naturally, if a man is carrying sperms and one of the sperm is genetic material is mutant, that person may not suffer himself. But if that sperm with mutant genetic material participates in formation of a zygote, right, and if that mutation is compatible with life, then the baby which is born as a result of that fusion of abnormal sperm and ovum, that baby will have same mutation in all the cells. Again, let me repeat it, recap it. I'm saying that let's suppose there's a man standing here and he develops mutation in his one of the sperm, and unfortunately the same sperm participates, you can say fuses with an ovum and results into formation of zygote, and that is zygote eventually becomes a full organism, human being. Now, you know, because sperm contributed defective genetic material to the ovum, right? So naturally zygote had one defect, genetic defect, and if that was compatible with life, when every time zygote will divide from 1 cell to 2 cell and 2 to 4, 4 to 8, so on and so forth, what will be the result? The baby which is born, in every cell that genetic defect will be there. So it's quite possible that baby itself may have inherited disorder. Number one. Number two, if again that defect is compatible with life and compatible with reproduction, that baby grows up, even though that may be abnormal baby or that may become abnormal adult, that grows up and reproduces. Now this person, for example, this is a baby boy, when he will be making sperm, now there is a high chances that the same defect will be passed to the next generations with high frequency. Is that clear? In the same way, we can take another example, that if there is a lady, suppose 2000 years back, there was a lady and there was an ovum present in her ovary, and that ovum has been hit by the radiation, right, and some genetic defect occur in a particular ovum. Unfortunately, that ovum participated in formation of zygote, and again that zygote was abnormal but compatible with life. So naturally the baby which was born by that woman 2000 years back, do you think that will be normal genetically or abnormal? abnormal, and every cell or only germ cell of the baby, or every cell of the baby. So because every somatic cell has a defect, so baby may have problems himself or herself, or he or she may pass the defect to the next generation as well. So we can say that when somatic cells suffer with mutations, somatic cells themselves suffer and their progeny suffer, but this problem will not be passed to the next generation. For example, this young man, if he has a defect in one of his somatic cells, he may suffer himself, but do you think he will pass this defect to the next generation? No. But if you are passing some mutational defect to the next generation, then it means that defect must be present in your germ cells. This concept is clear. Now we know what are mutations, and we know that what happens when mutations on the somatic cell. Few classical examples are like congenital defects and carcinomas, but such mutations cannot be passed to the next generation. Opposite to that, when we talk about mutations in the germ cells, then mutant germ cells may take the problem to the next generation, and so on and so forth. Any question here? Is there any question? There is no question. Now we come to detail of the mutations, but before we really go to detail of the mutations, I would like that we should have a very clear concept about the genetic code. First we should understand what is genetic code, normal genetic code, what are codons, what are anticodons, what are the general principles related with the genetic code system, right? Then we will come back to the mutations, because first we should understand how normal replication occur, we have already discussed. How normal transcription occur, we have already discussed. We should have some idea how the translation occurs, right? And then how defect in DNA may lead to alteration in RNA, and how change in RNA may lead to alteration in protein, and how change in protein may result in structural and functional abnormality of a particular cell, right? Now, let's talk about genetic code. All of you know that master plan for the life is present in DNA. If DNA multiplies and come back to DNA, it means this is replication. And if DNA leads to formation of RNA, then it is, yes please, transcription. And if messenger RNA is decoded by transfer RNA and a new proteins are made, then or peptide chain is made, what is this? Translation. Is that right? Now, we have to see, because to understand the mutations, we must know how changes in DNA may result changes into RNA, and sometimes they may result into changes in proteins, and that may lead to functional or structural alterations, right? To this whole series, to understand that, we must know what is genetic code. Genetic code is basically master plan of the life which is present in DNA, and this message is written by the triplets of nucleotides, right? Triplets of nucleotides. For example, as you know already, that DNA is made of basically 4 bases, that is guanine, yes please, cytosine, adenine, thymine. Is that right? Now, out of these 4, if we can combine them in different fashions, we can end up into triplets. For example, if we use the guanine, we can make GGG, this is one triplet. We can call it one codon as well. Is that right? It's a special code for a particular type of message for the protein synthesis. Now, or it may be GGC, or it may be GGA, right? Or it may be GGT, and so forth. You can make a lot of combinations. And total combination which are possible, that when you have 4 basically alphabets and you are supposed to make special genetic word or triplets, every genetic word is triplet, right? And you have to make all the possible combinations of triplets out of these 4 words. The total triplet which result will be, yes please, 64, right? Total triplets which can be formed is 64, right? It means that how many codons can be made of different types? 64. Is that right? Again I will repeat that what is codon, right? Codon is basically a triplet of nucleotide which classically appears in RNA, but of course it is dependent on the code system present in DNA, right? So primary code system is present in DNA. RNA is just a functional copy of DNA which comes into cytoplasm, and there it is translated into protein sequences. If there is amino acid sequences and protein formation, if this RNA is messenger RNA, right? Now, when we are talking about that, we are having how many basic alphabets? 4 types of nucleotides in the DNA, and we are making possible combinations by having 3 of them, any 3 of them together. So these triplets are codon system in the DNA and then codon system in RNA, right? Their complementary codon system is made in RNA, where in place of thymine we use uracil, and in place of deoxy sugar we use ribose, not deoxyribose. In RNA we use ribose. Now, out of these 4 combination, 4 alphabet, when we combine them in different ways, the maximum number we can end up into 64. Out of this 64 triplets, 64, okay, we call it triplet, people will call it codon, codon in DNA and then ending up into codon in RNA, right? We simply in our discussion we call them triplet just to keep the mind clear. 64 possible triplets can be there. Out of these, 61 triplets are meant for coding of, yes, they are meant for coding of, yes please, amino acid. A particular triplet may be meant to signify for a particular amino acid. But out of this, 3 are present as stop codon, as stop codon. Now let me tell you what is really meant by the stop codon. In the previous lecture, we discussed about a typical structure of messenger RNA. We said that in messenger RNA, suppose this is the 5' end. The 5' end, you remember there is guanosine, methylated guanosine, right? And here it is, this is 5' end. This is a mature messenger RNA. This is 3' end and here is attached, yes please, poly A tail. You remember this basic structure of a mature messenger RNA. This is 5' end, that is 3' end, right? And this is leader sequence which is not translated. This is the trailing sequence which is not translated. It means this is the area which should be translated into protein. Now, the point from where protein synthesis should start, there is a very special type of, you can say, codon, that is usually AUG. When in the sequence AUG appears, this is the point from where ribosome will start translating, and opposite to AUG, the transfer RNA will bring, what they will bring here? Methionine. And in case of bacteria, they will bring formyl methionine. And in case of eukaryotes, this transfer RNA will be carrying methionine. Actually, this is the point from where this codon AUG, suppose this is adenine, this is uracil, and this is guanine. This AUG means here there should be inauguration of protein synthesis. There should be start of protein synthesis. You can remember it like this. What are the spellings of inauguration? So you have to remember it. That start codon is inauguration codon, the point from where protein synthesis will be inaugurated, right? And this point from where inauguration or peptide synthesis will start, it is AUG. Of course, this is one of the 61. This is one of the 61 because it is coding amino acid methionine. Is that right? So it is always out of this one, yani AUG, out of 61, AUG is always working as a start codon. Now, what is the concept of stop codon? Actually, what really happens from inauguration point, from AUG, as ribosomes will start moving forward, right? For every 3 point, they will bring a transfer RNA, and that transfer RNA will bring a specific type of amino acids, and in this way amino acids will be placed into right sequence so that ribosomes can make a chain of the amino acids, and we should end up with the formation of peptide chain. But at the end of the system, there must be a point where there should be termination codon or stop codon. In some genetic language, it must be written that here no further amino acid should be incorporated or added, right? This is the point where we have to stop. Is that right? As in the end of the book, it is written the end. Sometimes in the end of the movie, it is the end. Sometimes in the life, right? So, in the same way, in the end of messenger RNA translating unit, here is somehow written the end. No more amino acid for the peptide system. But the end is not written in English or any other language, it is written in genetic language. In the genetic language, there are 3 ways. There are 3 types of codon, which may be, any one of those 3 may be present here, and that gives a message to transfer RNA that don't bring any more amino acid, right? Now, what could be those 3? Number 1, that may be you go away. It's saying to the peptide system, you go away, no more amino acid, you go away. Or it may be, there are 3 choices, stop codon. There can be 3 types of stop codon, right? You go away. Or it may be you are away, or very good, you are gone. It's just like that lady who is disappointing some young man. First she says, you go away. Then she yelled at him, thank God you are away. And then she called him up, you, right? You are gone, maybe to the hell, I don't know. But this is what is happening at the end of translational unit of messenger RNA. So whenever in the messenger RNA, any one of these 3 points, 3 codon appears, that signifies stop codon. That signifies stop codon, right? So what did we learn up to now? We are talking about the genetic language, because first we should concentrate what is the genetic language, then we'll say how there are abnormalities into that language, and then we'll end up that how those abnormalities may end up into mutations and what are the effects of mutation, right? Let's come back. We are talking about that genetic code system is basically master plan is in DNA, and every coding point, every genetic word basically consists of how many? Triplet of 3 nucleotides, and these are transcribed codon system into RNA, and if it is messenger RNA, then they are sensed by transfer RNA, and according to that, what is happening? We are choosing amino acids, right? Now, in this particular messenger RNA, this may be one codon, this may be second codon, these may be third, fourth, fifth, sixth, seventh. These 7 codons are there. And when a particular transfer RNA come, let's suppose I draw here transfer RNA, right? Of course, in case of methionine, let's suppose methionine is an amino acid which is carried by this transfer RNA. This is transfer RNA which is carrying this methionine. This is the 5 end of the transfer RNA, on 3 end we are having methionine. Actually, how this methionine carrying transfer RNA will sense the presence of AUG? Answer is that it is having points here which are called anticodon. If this is codon, then this is anticodon, right? So actually, codons are sensed by the anticodons of transfer RNA, and when they are complementary to each other, right, a particular type of amino acid will be lining up into sequence. For example, if it is adenine, what should be there? Yes, please. There should be, yes, uracil, and in front of uracil there should be, yes, please, adenine, and in presence of guanine there may be cytosine. It means the transfer RNA which has anticodon of UAC is basically carrying methionine. So it means when in messenger RNA AUG appears, this messenger RNA codon has to match with UAC anticodon of a particular transfer RNA, and this particular transfer RNA is carrying methionine. So methionine will be put into place. Is that clear? Now, of course, from where AUG came? It came from DNA. When from the DNA, we will making RNA, right? Let's suppose this was the sense strand, right? So there should be nucleotide sequence here, right? And RNA was made complementary to this point. If this was the AUG, now you can infer, you are quite intelligent, you can infer what was really present in DNA. What was present in DNA? There must be T, A, C, TAC. So it means in DNA original codon was TAC in the template, and anti-template had A, T, G. Okay, let me make it clear before you really mess up the concept. Let's suppose this is the particular codon we are talking about, and this was having adenine, uracil, guanine, AUG. Is it clear? Now, what really happens that this particular codon in messenger RNA was going to match with a anticodon UAC of a particular type of transfer RNA, and that transfer RNA should be carrying methionine. So we can say in many ways, we can say methionine is basically carried by a transfer RNA with UAC anticodon, or methionine is coded by the codon AUG present in messenger RNA. But originally from where it came to the messenger RNA, this message, it came from DNA. So let's talk about DNA. So in the DNA we can make 2 strands. Let's suppose this is antisense strand and this is the template or sense strand or template. Now if this is the template, you can understand that what was really there? If it is AUG, so this RNA when it was being made, what was here? AUG. If this RNA was having AUG, this point, then it means what was present in DNA? Yes, please. TAC. Is that right? So this was the template. And in the anti-template, what was present there? Yes, please. ATG. Here you see very interesting thing. Okay, another thing I can show, just to, in the DNA, if here it is G, 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 what should be present in RNA? C, C, C. And what was on this strand? C, C, C. What you really see? You see that one strand of the DNA act as a template and complementary strand of RNA is made. And complementary strand of the RNA is more similar with the anti-template strand of the DNA. Am I clear, Dr. Saab? It's clear up to the end? Is that right? Then whatever is present over here, now look. If we bring a change at this point, it's quite possible the change in this triplet, right, may result in change into this codon. And then if this codon is changed, that may end up into, right. So we were talking about genetic code. Genetic code is primarily present in DNA. Genetic code is present in DNA. What is genetic code? It is a language, genetic language, in which triplets of nucleotides are used to signify particular type of amino acid to be placed in a particular sequence in proteins. So basically, you know, all life, major functions, structure and function of the cell depends on protein, and protein synthesis depend on RNA, and RNA synthesis is coming from DNA. It means master plan is in DNA, from DNA to RNA, from RNA to proteins. Then we were talking about how this genetic code goes. We were saying that basically we have how many bases? 4 bases, and out of these 4 type of nucleotides, right, every triplet should be having, of course, 3 nucleotides, right. If every triplet has 3 nucleotides, how many total triplets you can make out of these? 64. Then we were saying that out of 64, 3 were stop codon. Stop codon, we have already discussed, you go away. One stop codon is you go away. Then there is you are away. Then a heavy message, you are gone. Is that right? These are stop codon. Then we said out of these 61, all of them are coding for amino acids, but one of them was a start codon, that was AUG, which basically is a signal to place such transfer RNA which will carry methionine, right, in the beginning of the peptide chain formation. Is that clear? After that, we were discussing this point that basically DNA has a template and anti-template. Transcription is basically, you can say, transcribed RNA is complementary to the template sequence, right? And if this is complementary to template, as I told you, template is triple G, then RNA should be triple C, right? But actually, anti-template is also triple C. So it means RNA is, even though complementary to template, but it is more similar to anti-template. Is that clear to everyone? Now, we will come to certain other principles and rules and regulations related with the genetic language. We have already discussed the genetic language, every word has 3 nucleotides, right? Now, there are certain basic principles which observe all this information. Principle number 1 is, there is specificity. There is Specificity. What is the concept of specificity? Specificity means that a particular type of codon will always signal insertion of a particular type of amino acid. For example, let's make a diagram where we will analyze the principles of genetic code. Suppose here is DNA, right? Now, if it is AAA, let's suppose it is AGC, here it is TAG. Now, if this is the starting point for message or master plan, this should be one codon, this should be the second codon, this should be the third codon. Is that clear? Now, when we will make out of it, let's suppose such RNA which is going to translate naturally against the AA, what will be making? U, and here it will be, yes please, U, C, G, and in third case it should be A, yes, U, C. Now, these were codons in DNA, right, in the template. Now, they have been transcribed in complementary codons present in RNA. Is that right? Now, when this RNA is undergoing translational process, this is suppose RNA, right? Now, what really happens? There should be a transfer RNA which should be able to recognize this codon of RNA. Now, what should be here? For U there should be, yes, A. What should be here? A, and ideally speaking what should be here? A, right? Let's suppose if this is the anticodon of transfer RNA which is able to complement with this particular codon, one codon, second codon and third codon. And it means whenever there is U, transfer RNA which is coming, right, there are chances that it should have AAA. And whenever it is AAA, let's suppose it is carrying an amino acid, let's suppose amino acid, we are just supposing, it may not be true, let's suppose lysine. Or we can make it just amino acid, there are 20 amino acid, let's suppose number 1 amino acid. So in genetic system, whenever messenger RNA will have triple U, right, transfer RNA will match with triple A, it will be carrying amino acid number 1. Is that right? But actually, you see, look here, the beauty of the genetic system. Template eventually is complementary to the anticodon. Template is eventually complementary to the anticodon. And messenger RNA codon, similar, yes, again let's repeat it. This was template and here we'll put anti-template. What was anti-template? E, T, T. Is that right? Now, what is happening? It is triple A suppose codon in the DNA. Then it will be transcribed into messenger RNA codon which is triple U. It will be sensed by transfer RNA anticodon triple A and suppose it is meant for amino acid number 1. Now, if a similar follow further, that if it is the next codon is DNA codon is AGC. So naturally the complementary codon in what is this? Transfer RNA should be UCG. What should be ideally speaking here? Yes, please. In front of you, A, G, C. And whenever A, G, C is there, it is amino acid number suppose 7. Right? In the same way, when there is TAG, codon in the messenger RNA AUC, then anticodon will be there. Yes? Is it T? Yes, U. Then A, G. And what could be here? Let's suppose amino acid number 3. Now, let's concentrate on one thing. What is the relationship concept of specificity with this particular discussion which we have made? The concept is this, that whenever in DNA triple A will appear as a codon, it will always mean amino acid number 1. It is always a message that what should be inserted there? Amino acid number 1. If this codon, DNA codon is properly transcribed into RNA codon and RNA codon is properly just translated, there should always be amino acid number 1, not amino acid number 7 or 3 or 10 or anyone else. For example, if amino acid number 1 is isoleucine, there is no way that there should be tryptophan here. Am I clear? It means that this particular codon, right, in DNA or codon in RNA always signifies a specific type of amino acid. Is that right? Let's repeat this example with another thing. When it is TAG, codon in the DNA, in RNA it becomes AUC. Is that right? And eventually it will be matching with such transfer RNA which will be carrying amino acid number 3. Let's suppose it is histidine. Right? So it means that whenever in the DNA, in human DNA, wherever this triplet appears, amino acid which is coded is always number 3. It means a particular codon in DNA and eventually a particular codon in RNA is very, very specific for particular type of amino acid. This is called specificity, the concept of specificity. Is that right? It is not possible that in chromosome number 1, TAG means, for example, there is a gene in chromosome number 1. If in a gene chromosome 1, TAG mean, suppose amino acid number 3, it is not possible that in chromosome number 10, somewhere TAG should mean chromosome other than 3. Right? So what did we learn? That a specific codon in DNA we transcribe into specific type of RNA codon is always specific for a particular type of amino acid. Is that clear? No problem up to this. Then there is another interesting point, right? That this principle is not only specific, it is universal also. Truly speaking, it is not universal, it is almost universal. This language is universal. The second, universality is there. Universality is there. What is meant by universal language? Genetic coding language is universal. Not only this language is specific. Now what is our concept of that it is universal? Let me tell you one thing. Let's suppose that if TAG in human beings, in human DNA, if TAG eventually results in amino acid number 3, you will be surprised to know even in monkeys, even though monkeys are very different from you, at least to some extent, in monkeys DNA, if TAG appear there, it is again meaning 3. Even wife of the monkey, if she has DNA with TAG, she will also end up message for amino acid number 3. Okay, you go lower down, we talk about fish. In DNA of the fish, if TAG appear, again the same amino acid. Even you go to virus, look at it. You are such a complex human being, other side a humble virus. like HIV, even though it is very nasty, right? Or any other virus, right? If a virus has DNA virus, suppose, not RNA virus, let's suppose DNA virus, of course HIV is RNA virus. There's some DNA virus which has a message TAG. So which amino acid will be there specified? 3. Now we come to know that this system is not only the particular codon is specific for a particular type of amino acid, rather this coding system is universal in the biological world. Same in the virus, same in bacteria, same in human being, with very few exceptions. With very few exceptions. For example, some exceptions are seen in mitochondria. That is, some of the mitochondria. In mitochondria, there are some codons which are coded in a different way, right? Is that clear? They are coding a different amino acid than others. It means the genetic language, when it evolved accidentally during evolution, the genetic coding language evolved by accidental way during evolution, then it was never altered. We say this is a accident frozen in time. This is just a happy accident or sad accident? Happy. No, I don't know, right? But I know it's there, right? So this is a genetic language which was somewhere made during the evolution. I don't know by virus or by bacteria or who, but once it is made, it is being. Now this is very strange. Human beings talk in so many languages, but DNA talks only one language. Even you see, dog barks different than some humans. But DNA of the dog and DNA of the human is coded and decoded in the same fashion in many aspects. This is also an example that system is, genetic code is universal. It is universal. Is it clear? Now, genetic language is specific. A particular codon specifies a particular type of amino acid, and this is universal. Universal means that this code system is evolved in very early stage of evolution. Right. So we were talking about the third special feature of genetic code, and third feature was that genetic code is showing redundancy. Now we have to see what is our concept of redundancy. In literal meaning, it means genetic code is redundant. Redundant means it is bit extra. It looks as if it is more than what is required. How come? Look here. The total codons which are coding for amino acids are 61. The total genetic codons which are coding for amino acids are 61, but amino acid to be coded, amino acids to be coded, they are only 20. It means that amino acids which should be coded are 20, but codons are 61. It means one amino acid may be coded by more than one codon. One amino acid may be coded by more than one codon, but still a particular codon is kept specific for a particular type of amino acid. Is that right? This concept is called that genetic code is redundant. There are multiple reasons for that. One interesting reason is that let's suppose TAG result into RNA with AUC, and what should be the amino acid? 3. Now another way to code the same amino acid is another codon. That next codon is TA, let's suppose TAA. So naturally it should be what here? AUU. Now here it is AUU. Now you know that messenger RNA on this end this is 5 end, and on that side this should be 3 end. You know always genetic system is read from 5 to 3. It means this is nuclear in this particular codon, and this particular codon, this should be considered nucleotide number 1, 2 and 3. It means when we read messenger RNA for this particular codon, it should be AUC, right? But when we go for transfer RNA, let's suppose I show you the transfer RNA. This is the 3 end of the transfer RNA. This is the 5 end of the transfer RNA, right? And ideally speaking, what should be here? U, A. Ideally speaking, they should be A. U, A. But you see, where is the 5 end of this? Okay, I have made a mistake here. This should be the 3 end, and here it should be 5 end. Please correct it. And here is the amino acid carried with the 3 end. Now listen carefully. Why I have made this change in my diagram to correct it? Listen why. Because they should be anti-parallel to each other, right? If it is from here to there, it is 5 to 3, then it should be in this direction 5 to 3. Is that right? The backbones of both sides should be anti-parallel. If this is 5 end, then this cannot be 5 end, this has to be 3 end, and amino acid is carried on 3 end, right? Now, it means for this particular RNA, messenger RNA, this is 5 end and this is 3 end, but in this case, this should be 3 end and this must be 5 end. When you will read the codon, that is AUU, but when you will read the anticodon, it should be read again 5 to 3, not UAA. Anticodon should be read as AAU. Is that clear? It means that in case of when the codon of messenger RNA and anticodon of transfer RNA, they are matching with each other. Let me repeat. When codon of messenger RNA and anticodon of transfer RNA, they are matching with each other, they should observe the principles of complementarity of the nitrogenous bases, principles of complementarity of nitrogenous bases, and their principle of backbone should be anti-parallel should also be observed. Is that clear? But there is very interesting situation here. We are saying that nucleotide in the codon of messenger RNA, nucleotide number 1, base number 1 match with the base number, and with any anticodon base number 3, and base number 2 match with the base number 2, and base number 3 in messenger RNA codon adjust with the base number 1. Is that clear? Now, I will repeat it, then we'll go to a slight change over here. Number 1, the rule is this: when messenger RNA with codon and transfer RNA with anticodon, when they are adjusting with each other, the rules are: number 1, backbones should be anti-parallel, and number 2, nitrogenous base should be complementary, right? In this case, nucleotide number 1 of messenger RNA has to be complementary with the nucleotide number 3 of transfer RNA. 2 of messenger RNA must be complementary with the 2 of transfer RNA. 3 of messenger RNA should be ideally complementary with first of transfer RNA. As you know, in real life, ideals are not there, and usually we settle less than ideal. Same is true about transfer RNA also. And messenger RNA. Codon and anticodon system, when they are matching with each other, right? Again, codon of messenger RNA and anticodon of transfer RNA. when they are matching with each other, matching with the first and the second of messenger RNA should be absolutely traditional. Adenine should always be with uracil. Uracil should always be with adenine. If it is here guanine, it must always be cytosine. And if here is cytosine, it should always be guanine. Listen carefully. What I was talking about. Ideally speaking, what should happen? That all three bases of the messenger RNA should be fully complementary with the three bases of transfer RNA, or we can say all three bases of anticodon should be absolutely complementary to the three bases of codon of messenger RNA. But as it happens in real life, it happens in genetic coding system also that ideal situation is not met with, and then as in practical life we settle less than ideal. Is that right? In the same way, transfer RNA also sometimes settle with less than ideal complementarity situation. For example, one should always match with third of this. First of messenger RNA should match third of this. This should be traditional matching. Second should match with the second of codon should match with the second of anticodon. It should also be strictly traditional. But third case, attention please, may be complementary, may not be complementary in a traditional fashion. This is the point which I want to highlight. Third, in codon, the third nucleotide may or may not be truly complementary or traditionally complementary with the first of transfer RNA. What I really mean by this? It is just like that, as you have three demands. For example, look at this young man. He has three demands. He says he wants to marry a woman. Secondly, he wants to marry a very pretty woman. Thirdly, he wants to marry educated woman. Right? You have three demands. You want to marry a lady, female. You want to marry a pretty lady, and third demand is she should be educated. But if you don't you find a person with all three things, you may settle with two of these three things. I hope you will not settle for some abnormality in the first case. You want traditional matching. In second example, you would also, for example, you say, I want definitely lady and very pretty. You say, fine, if she is not educated, no problem. I will settle with the less educated. It means out of three demands, you are willing that third demand may not be fulfilled properly, but first and second should be. Is that clear? Same is true over here. This is coming with the demand. That demand between first should be traditional, second should be traditional, third may or may not be. For example, in front of you there should be A. Or sometimes, now see the real adjustment at the end of, let us suppose this is star amino acid. It's quite possible it may be U, A and C. Do you think normally C can adjust here? No. But if this anticodon and transfer RNA adjusts with this, it means first and second are strictly A will go with U, U will go with A, but this U should not ideally go with C. And if it goes, then it is traditional matching or non-traditional? Non-traditional. Am I clear? Actually, it happens in codon-anticodon system that as far as messenger RNA is concerned, codon first and second should be strictly complementary to the third and second of anticodon. But third of the codon may or may not properly match or complementary in a traditional fashion with the first of anticodon. Is that right? Now what will be the result? But there is a beauty in genetic language. Even though this transfer RNA can, suppose this is transfer RNA number 1, this is transfer RNA number 2. Now with this particular codon, One can adjust as well as two can adjust. The beauty of the system is if one is carrying star, two should also be carrying star. So that still amino acid is the same which is specified over there. It means star amino acid is coded in more than one way. Star amino acid is coded in more than one way. Is that clear? Right? This concept is called wobble hypothesis. This concept is called wobble hypothesis. Wobble hypothesis is that when you are matching codons of messenger RNA, when you are matching codons of messenger RNA and anticodon of transfer RNA, as far as messenger RNA is concerned, first and second nucleotide bases should adjust with the third and second of anticodon in a very traditional fashion. Third may or may not adjust. Is it clear? So all this example, and as I told you previously that total codons are 61 and total amino acid to be coded are 20. So even though a particular codon is always for a specific type of amino acid, but some amino acids may be specified by more than one codon. For example, let me repeat it, that if AAA, it goes for amino acid number 10, let's suppose,